Hello, everybody. I will be speaking about the complete oligo synthesis masterclass today, going from the chemical cycle and continue into how you should set up the laboratory. So, agenda for today would be the oligosynthesis cycle, general reagent need, which includes solid support columns, phosphoramidites, ancillary reagents, specialty materials. Then some of the reagents used by techniques, I will pick a couple. Then next would be picking a correct synthesizer model for your research or your production, necessarily chemicals, space, ventilation, equipment in laboratory during synthesis. Also, I will be speaking about post-synthesis reagents, equipment, and accessories, and other considerations you will need to know before setting up the lab. So let's start. Oligosynthesis cycle, some of you may be very familiar with that and some might not be. So what the synthesis cycle here shows is a standard addition of an oligo base per synthesis. This is for just a single base. So where we start with your CPG or polystyrene on and a base attached to it. So the first base could be DNA, RNA, linker, any sort of a modifier. This is your 3' end. The glass bead has a linker, LCA linker attached. So first step, what we would need to do is to activate the 5' OH group by removing this protecting group. It's called DMT. To do that, we add acid. It's called a D-block reagent. There is a different concentration, different combination of this chemical. It's a blend of two, usually goes consisting of DCA, TCA, in toluene or in dichloromethane. When the chemical hits the bead, this bright orange color comes off. It's called tritol. What it does, it activates this OH group for the next addition. Next addition would be putting the second base on, which is this is your cyanoethyl group. This base gets mixed with activation, which could be ETT, BTT, or DCI. And then during this reaction, it's called coupling, the two bases are added together. So the following step would be capping of uncoupled bases. This reaction is not 100%. So what we want to do is we want to kill any available OHs that's left after this reaction. So by addition of two reagents, cap A and cap B, you will actually block this available OH. So this reaction is dead. It's not going to continue forward. The reason why we do that, so it doesn't accumulate a lot of impurities. So the next step will be the oxidation or sulfurization, depending what you're trying to achieve. The reason why we do that, if you can see, there's a phosphate here with only 3 bonds. Phosphate is a penta. So 3 bonds is unstable. So what we want to do, we want to stabilize it and make the phosphate connect to 5 bonds. By adding oxidizer, it introduces an oxygen, double bond here, or in case of sulfurization, it would add a sulfur here, creating a thioate. So now, as you can see, we have now 2 bases added to the bead. And after this step, we go back to removing the D-block group. What is TPG, or a controlled pour glass? It's a little shard of glass. It's non-swellable. It does have this little grooves in it. This is a part of the process to increase a surface of the area. The size of the grooves come in different width. So they call it the pore size. They come in 500, 600 angstrom, which are good for oligos that are under 35 mers because the hole is very small and it fills up. What you don't want to do is trying to make a really big molecule and stuff it in a very small space, because then what you're going to do is you're going to kill the synthesis, because the flow through those little channels is going to be constricted. So here's a little guide to the sizes for the size of your oligo you want to make. So 1000 angstrom, you can do up to 70 mer, 2000 angstrom up to 130 mer, which is good for CRISPR application, guide strands. And then 3000 angstrom, you can go up to 170. There is a different solid support. It's called polystyrene. That is a bead that's coated with a linker. So it's like, imagine a little ball or a little hedgehog that kind of curled in on itself. The difference between the two, polystyrene only has a surface area. So when you trying to make a really long oligo on it, it's not going to happen. At some point, the chain is kind of going to fold over and won't allow the next base to couple. So you will see a drastic drop in your coupling efficiency. So polystyrene is good for ASOs, large scale, because the coverage and actually loading on it is much higher than the CPG. So both supports have a good space for a specific applications. So phosphoramidites, this is your basis. This is what you build your oligo out of. Imagine making a little necklace with the 4 different color beads, and you put one bead on at a time. So you have either RNA or DNA-based phosphoramidites. The one here is thymidine. So this is a DNA. For the RNA-based, you will have a molecule attached to the 2' right here. So it will be either OH, which is a basic RNA. You can do 2' methyl, fluoro. There is a different modifiers as well, like linked nucleic acid or unlocked nucleic acids. So the basic 4, 5 amidites is thymidine, adenosine, cytidine, guanosine, and uracil. Uracil is the RNA, thymidine is a DNA. So also on the base that is attached, this is where you're going to have your ACGTU. You will have different protecting groups. The reason why, because the chemistry you're putting the oligo through is pretty harsh. So what you want to do is keep this particular base attached to the ribose. Ancillary reagents. As I showed you through the cycle, we do need certain chemicals to push the reaction through. So acetonitrile is a single base solvent. It is used to wash between the steps and as a diluent for dissolving phosphoramidites. D-block, which is the acid. So it does have multiple different variations, but what it does, it removes the DMT group from the growing strand and allows the next base to be added. Activator, it is mixed at the same time with your phosphoramidite solution, activates the phosphoramidite. It's not a part of your reaction, it's just like an intermediate. Capping solution, it does use the block active sites, so you do not build an N-1 ladder. And the oxidizer used to add oxygen to the backbone. And also sulfurizing reagent, as we talked about. So NAC product portfolio, as you can see, there is multiple applications using phosphoramidite chemistry. So, for example, the cell delivery and uptake is lipophilic modifiers, vitamin modifiers like vitamin E, and GalNAc modifiers. What it does, it actually helps the molecule to be delivered into the system. Chemical phosphorylation. The phosphorylation reagents are good because for currently the gene synthesis is a pretty big thing in the industry. So what you do, you add the phosphorylation reagent to the end of your oligo, and it helps it with the help of an enzyme to connect the two strands together. Then there is colorimetric detection, conjugation, electrochemical detection, fluorophores and quenchers, which is right now is quite popular because of the COVID testing or in MDX field. So you have your black hole quenchers, blueberry quenchers, Tamra, and you have multiple fluorophores that go with it. Then you have specific modifications for nucleus resistance, the structural studies, backbone modifications, branching modifications, photocleavable modification. That's an interesting part, that you actually use light to deprotect, to remove the protecting groups, and spacer modification. This is for extensions or for branching, so you can then label the oligo or connect it or conjugate it to something else, like a surface, to the peptides, to the proteins, etc. So commonly used reagents in molecular diagnostic oligos. So colorimetric detection and capture, conjugation, fluorophores, photocleavable, and spacer modifications. Example of diagnostic oligo techniques that oligos are used in is, as I say, quenchers, aptamers, qPCR, RT-PCR, binding assays. So who works in the MDX field will be using this type of techniques for the oligos you're going to be making. What is the quencher? This is something that people constantly ask. Quencher is something that is when paired up on a specific distance with a fluorophore, kind of shadows it over. So fluorescency is reduced or non-existent. But when you separate the two, the fluorescein lights up. So this is a very commonly used technique in diagnostics. So if you have your oligo that kind of folds like that, you have quencher here and the fluorophore here, it doesn't fluoresce because they're close and they kind of like quencher covers the like the fluorescency. When you add it to your assay and it finds a match, like for example, the positive COVID presence, so the oligo actually unfolds and sits on top of each other. Hence, it separates the quencher and the fluorophore, and then you will see the fluorescent reaction. If the template is not there, the oligo is going to stay closed, and you're not going to have a positive result. This particular technology just uses a standard DNA phosphoramidite, ACGT. It uses the fluorescent phosphoramidites, such as fluorescein, hex, ROX, Tamra, cyanine 3, cyanine 5, and it uses a CpG with a quencher molecule. Also, there is some new techniques to increase the signal and reduce the background when you can actually add an internal quencher into your sequence. So you will have quencher, DNA, quencher, fluorophore. And for different application or different pairing of fluorophore and the quencher, you will play around with a different length of your oligo as well. But those oligos are around no more than 30 mers, 35 mers. So this is easy to make. And as I say, having your own capabilities in the laboratory and not wait for weeks and weeks and months of getting something from oligo house actually has been very beneficial during the last few years for people. So there is a big application for the therapeutic oligos. Those of you who are in siRNA world, in the ASA world, which is an antisense technology, it's a quite wide field of applications and a lot of different modifications that go into this field. Also, the oligo buckets that apply would be cell delivery, conjugation, modifications for nuclease resistance, because when you do RNA, RNases love it. So what you want to do, you want to block it for the RNases not to recognize it in the human body. Backbone modifications like thiolation and spacer modifications for conjugation purposes. So example of therapeutic oligo techniques is antisense, which is one of the oldest ones, mRNA, siRNA, exon skipping, aptamers, ADAR, and one of the latest technologies, the CRISPR, which is gene editing. For example, CRISPR is when using used with a Cas9, which guides to recognize and cleave specific DNA complementary to the CRISPR sequence, used for gene editing to get the desired result. So if you have a mutation, genetic mutation, that could be fixed by using this technique. Guide strands are usually about 120 bases long, which is not an easy synthesis. There are some tricks and tips for that. And it also uses different RNA or 2'-methyl phosphoramidites. Due to the length of this particular string, you would want to use 2000 or 3000 angstrom CpG, for example. So every technology we do introduce has a specific recommendations for what CpG to use, what angstrom, what reagents, and any kind of additional tricks to synthesis cycle, modifications, and how to make it. Okay, gene synthesis. It is applicable in many fields right now. The most talked about one right now is the gene synthesis as a template for the mRNA. However, this is something more current, but the gene synthesis has a very long history and quite interesting development. What put gene synthesis on the upfront of everybody's eyes was the Duke University poliovirus glioblastoma treatment. The scientist cannot use a live virus to cut it, religate it, and inject it because it's not 100% reaction. So the gene synthesis technology gives the scientists right now the capability of designing a gene, putting it together while removing any kind of dangerous bits out of it. So for poliovirus, what they did, they removed the replication cycle from it, like pretty much castrating the virus. And also the recognition side where poliovirus infects a healthy brain cell. What they did modifications on it so it would only recognize a cancer cell. And glioblastoma is a deadly cancer. Your survival rate is 0, and your life expectancy from cancer starting is about 6 to 9 months, and then you're dead. It's a very aggressive cancer with no cure. However, using gene synthesis technology combined with the virology and how the virus attacks the brain cells, Duke University was able to create a virus that only eats the cancer. And after it eats it, it goes dormant because it can't replicate. What they do, they synthesize it with specific modification, and then they inject it into the middle of the brain tumor. And currently, they just surpassed 3-year remission, which is great. So, very passionate subject for me. But back to the gene synthesis template for the mRNA. So the way you do gene synthesis is you take your full sequence, you cut it into little shorter fragments that's manageable to make. Then you do a second strand, which is the antisense strand or reverse strand, and with the little overlaps, cut it in different position. So when you're trying to assemble the gene right here, so this is going to be your forward oligos, those are going to be your reverse oligos. As you can see, they overlap. So what you do, you put a 5' phosphate on it, on each oligo. And then when you put them together, you add ligase. And what it does, it actually puts longer pieces together, until you actually have a full length gene assembled, or a template in this situation. Then you can amplify it, clone it, and find the perfect clone for your target. After that, you can infect the cells, or you can cut it, put it in a different vector, infect the cells, and it will start producing the mRNA because the vector would tell it to do so, like a cell machinery. As I say, this is quite a old technique, but it's one of the most relevant right now. So now I touched a little bit on different applications. Of course, there's many more. And, we can maybe go over that at the different class. But right now, what I want to start talking to you about is how to set up a lab if you want to bring your oligo needs in-house, not just order it from the vendors, but actually have a control what you want to do. A lot of people want it to be for the fast. Some people want it to have for the proprietary, so you don't have to send your sequences out. And for that purpose, there's different sizes of equipment that you might consider. So our instrumentation line consists of different models and a different throughput. So if you are an academic and you want just to make a couple of primers here and there, you might want to consider our Mermaid 4. What it does, it only has 4 positions, small scale, doesn't use a lot of reagent, and it's very compact and can find, you can always find enough space in your hood to put it. Other machines that we do offer, it's for different applications again. So our Mermaid 6 and Mermaid 12 synthesizers actually can go up to 200 micromole per column. Makes it really good model to do synthesis for my study, PD study. It's a mid-scale synthesizer, but it also can do a small scale as well. It's a very diverse or very adaptable instrument. Then we're going more into higher throughput instrumentation. This is more for screening or for if you do have multiple oligos you want to make a day. So we have the mid-scale, which is our 48 synthesizer, and we also have 3 different variations of plate synthesizers, 96, 192E, which is a high throughput and actually featured right here, and our 192X. The difference between the instruments is number of amidites. When 96E has 1 plate and 12 amidite positions, 192E has anywhere from 4 to 8 amidite positions, but 2 plates by 96. Those 2 instruments are good for primer synthesis, or if you have like an MDX to do a fluorophore and the ACGT. Our Mermaid 192X is one of the first instruments that's ever been developed. It can go anywhere from 10 to 64 amidite positions. And that particular instrument is really good for ASO screening, because then you can load all kinds of different RNA modifiers on your instrument and mix and match them in the same sequence. All cycles for DNA, for example, a 20-mer primer will take up to 3.5 hours total. It doesn't matter if you're making one oligo or 196 oligos. The speed is determined by how long it takes you from the D block to the D block. So the chemistry, as I said, there's different combination of chemistries for each reagent. And people usually use it either availability of the mixture, application, because for some techniques, some are better than the other. So it actually keeps the yield higher. For example, your BMT or BTT activator is very good for the long RNA. So in case of CRISPR, this would work better than the standard 0.25 molar ETT. For the D block, as you can see, there's different percentages. So you can have 3% DCA or 3% TCA, which is dichloroacetic acid or trichloroacetic acid in different solvent, either DCM or toluene. Also, you can go up to 15% of your acid. However, you have to be careful with it because acid is extremely strong and it does affect each base differently. So if you use a really high percent for your DNA, for example, you will see a lot of depurination and deamination on your bases, hence creating the mutations further down. So activator, there's 3 types: ETT, BMT, and DCI. DCI is mostly used in the synthesis on the chip or very kind of microfluidics because this particular activator doesn't crystallize. The oxidizer, there is different combination of oxidation. There is 0.02 molar iodine in water. There's 0.1 molar iodine in water, which is the highest. Then there is 0.05 molar iodine in pyridine or different solvents like THF, pyridine, water. Water is important for the oxidizer because that's where it gets its oxygen in a combination with the iodine. The color of this reagent is kind of like a color of coffee. So, and there's a combination of a cap A and cap B. So as you can see, different mixtures for each one, but you need to make sure that 2 of them are compatible, that you have the NMI and the silicon hydride in 2 separate bottles, because the combination of those 2 reagents is what creates the capping. So if you mix them 2 together, it's not going to work. You have to cap it separately, but on the instrumentation, they're added together into the column. So how does the instrument mermaid makes the oligo? So here's the 2 pictures of 2 different instruments. This is the picture of the M4. So you can see there's only 4 columns here. And this is the picture of the inside of M12. As you can see, columns are all different sizes. You can do this all in one run. So you can make it to 100 micromole. At the same time, you can make 15 nanomole synthesis. And you don't have to run separate syntheses for each of the sizes. So how the instrument works, there is a slide that you put your columns into. There's a line underneath the slide that goes either to tridal monitor or through the waist. And the slide goes back, forth, to right, to left. And in the case of our 2X synthesizers, which is 48 and 192Xs, it's an XYZ synthesis. So it goes from every possible angle, kind of, and hence allowing much larger amount of amidites to be introduced into the injection head. So the injection head is stable. The only thing that moves is the slide. So the column moves under the injection tip. The instrument tells you, okay, release that many microliters of this reagent. The valve opens up. It's like a little shower head. It comes into the column, and then the vacuum pump pulls it through. This is where your CPG is. There's two little filters, and the magic happens in between. The coupling step and all the reaction step, except the wash, do have wait times. The reason why, so it actually has time to penetrate in those little grooves and give it time to react. If you just put it in there and flush it, you will never have an oligo. So the instrument programming allows you to set the specific time of contact inside the column. And also you put a lot of solvent in there, and it's slowly moving through, exposing the CPG to a fresh reagent. So spaces, clearances in your laboratory. Each instrument is pretty sizable. However, up to this point, you could put it into an 8-foot standard chemical hood. The reagent instrument does need ventilation. So unless you putting it in the hood, you can also put it on a bench or on the cart under a snorkel or a drop-down canopy. Something like what you have in the kitchen over your stove. So when you burn the eggs, the fire detection doesn't go off. So here's the width, depth, height, and the weight of each instrumentation. So ventilation, as I mentioned, this is the chem hood, and this is, and this is the snorkel. So it's kind of the arm that comes in from the ventilation. You can bend it. It has a little dish on it, and it would pull out the smell that comes out from the vent of the instrument and also help you when you put the reagent on. It's a pretty smelly chemistry. For us chemists, smell of pyridine in the morning is actually delightful, but for your office folks, when they're going to smell it, they're going to think something died. So you want to be considerate to your colleagues that do not work in the chemistry lab. So this is ventilation is important. So we do go over the requirements of your lab space and give you recommendations. The only instrument that would not fit in here, but would fit into walk-in hood, which is a ceiling to the floor, is 192X, because that one actually comes built in with the cart on the legs, and you can actually move it around. Gas supply. Each instrument does work with inert gas. There's 3 gases that are used in combination with oligosynthesis: nitrogen, argon, and helium. Helium used to be very popular because it used to be a very cheap gas, and it's one of the gases that doesn't make bubbles form in line during the synthesis. However, currently there's a huge delay or not enough helium in the world, and it's extremely expensive compared to the other 2 gases. So the first one I would recommend usually is argon, and we do recommend using it with the ultra pure quality, which is a very low water content. Something I forgot to mention to you: DNA chemistry is extremely water sensitive. You do not want your phosphate to be exposed to a high humidity or a high parts per million of water in your solvent, because it will kill your phosphate. So pretty much all the reagents and gas and conditions should be as close to low or anhydrous as possible. Hence, we recommend the ultra high purity. The reason is the gas pressurizes all your reagents, and if there's a lot of water in it, it would permeate into your solution, and your solution of the amidites and reagents would not last a very long time on the instrument. If you do, for some reason, cannot source the ultra high purity or low water gas, and you have to go with, for example, house nitrogen, a house nitrogen runs about at 300 parts per million. So what you would want to do is get one of those inline filters with a dry right in it. People who work in the lab, in the chemistry lab, fairly familiar with this substance. When it's dry, it's blue. When it turns wet, it turns pink. So that's the time to pour this out and pour a fresh one in. So this can be put between the gas tank and the instrument, and hence minimizing the amount of water in your gas. You do have to purchase a gas supply regulator. They're available pretty much everywhere. That's going to go control your gas flow from the gas cylinder into your instrument. So that's for setting up the instrument. Now for the oligo, after it's done through all the cycles, you have your little column, you have your little CPG. How to get your oligo off of it? What happens next? Because you can't just use it on the bead, though some people do for different applications. What you would need to do is actually, first step is to remove it from the glass. How would you do that? For example, for Mermaid 6, 12, and 4, there's this type of contraption that we provide. You put your tubes inside the contraption, you put your columns on the top of it, you close it, and then you add a reagent that would cleave it off the glass. That LCA linker will get cleaved, and your oligo is going to go into the tube below in your ammonia or AMA, which is the 2D protection solutions. After that, you would want to incubate it either benchtop or in the oven overnight for 2 hours, different temperatures. Again, for each type of chemistry, there is a suggestion how to do it to make sure that everything works and you remove all the protecting groups and you have a nice cleave off the support. As I say, post-processing chemicals, there's a list of them and there's different mixtures. The most standard one is concentrated ammonium hydroxide, or it's called a solution. Smells delightful as well, so please use it in the fume hood, not on a bench. What it does, you want to have it fresh, because if it's not fresh, it's been sitting. People sometimes get a 4-liter bottle to do 200 microliter reaction over 5 years. In about 20 opening and closing the bottle, the ammonia will actually exhaust itself. The percent will go down quite significantly, and your cleavage would not work. So you would lose your oligo, it would still sit on the bead, or your protecting groups are not going to be removed properly. So you will lose your yield and return on investment, so to speak. As I say, ammonia is readily available. It's cheap. You buy, I would recommend you buy smaller bottles, refrigerate them, and after opening it a certain amount of time, you dispose of it or send it to biology labs so they can do some buffers. So sometimes people use AMA. It is a controlled substance. And some companies need to have a DEA license to purchase it. However, AMA, for example, for the oligo houses, it's an extremely fast. So if ammonia takes 45 minutes to cleave, AMA takes only 15 minutes to cleave. And for some oligo houses, the faster you can deliver the oligo, the better. And also AMA is used in high deprotection or the easy deprotection amidites, which includes acetyl C and DMFG. So those are removed very fast compared to the traditional protecting groups. Also for the RNA, there's few things you would want to do. RNA is way more sensitive. So I wouldn't recommend using AMA on the RNA strands if you're doing siRNA, CRISPR, etc. Or if you use oligos to build the genes, because AMA does have properties that will damage the DNA or RNA in that situation. So for RNA, I recommend the very easy ammonium hydroxide ethanol 3-to-1 mixture, 3 parts ammonia, 1 part ethanol. Make sure that it's easy, lower temperature, and longer time. That way, it's much milder, the protection condition. It would not damage your oligo as much. So, and for RNA, there's also a secondary step if it's a traditional RNA, and it will have either TBDMSD protection or different types of protection on the OH, then you will have to do it for the secondary step. And you need to keep your environment sterile after that, because OH is where the RNases will attack and eat your oligo, destroy it. So after you've done with all this cleaving, deprotecting, now what you have left is your ammonia, the smelly substance. How do you get rid of it? What you want to do, you want to dry your oligo, lyophilize it, evaporate it. There's different terms for that. So what you need to do is to pick a drying equipment or lyophilization equipment to meet your needs. So if you're doing a 96 well, like high throughput, and you want it to dry fast, and it's not native RNA, you can use one of those zip evaporators that we have. This one gets hooked up to a line of house air. Air gets heated up, and the plate goes in, and there's pins go inside the plate, and they blow hot air. This is probably the fastest way to dry your oligo. There's a second choice. It's a speed vac. It's a centrifuge with the vacuum. You can purchase those. They're quite common as well. So in that situation, it spins, keeps the solution close to the bottom, and it lyophilizes the oligo, so it pulls the solvent out. So there are different ways of doing that. If you have questions on the specific ones for specific application or the instrument, I do provide different options for your specific needs. Quantification doesn't matter. You've been in the lab, you know what your 260 to 80 reader is. This little debris is a nanodrop, so this is for individual measurements. However, if you want to have a 96 well plates, there's different OD readers, your 260 to 80. My all-time favorite is Biotech. It's one of the small compact instruments, works really well. There's also different old ABI readers, etc. You can get one of those readers not just for 260 to 80. If you're doing fluorescency, there are some that do the dual wavelength, so it will read your concentration of a fluorophore as well. So after you quantify, you know, okay, I have that much micromole, nanograms, milligrams, ODs in my solution from one column of synthesis. What you do after that, you want to either purify it, analyze it, or just go ahead, precipitate it, and use it, especially for the primers. Just do a quick dissolve step. For QC and purification, a method to QC your oligos is recommended. There is various QC methods. I usually prefer to use 2 different ones. LC would confirm your molecular weight. However, the purity of the oligo could be done either by HPLC or by CE. The old Combicep instrument used to take the plate, goes in 96 probes, pulls everything out, and runs it in parallel. So in 1 hour, you will have a profile of your oligo and see how clean it is. HPLC doesn't do that. HPLC does one oligo at a time. So if you have a small throughput, like 4 to 6 to 12 oligos, HPLC is a good option. If you're going for the plate analysis, I would recommend you look into getting a Combicep. So, safety and other considerations. We're all working in a lab. We all have to deal with the fire marshal, with OSHA, with your own internal safety. And this is not just because somebody wants to, boss you around. It is really, really important because the reagents that you work with are hazardous, they're flammable, they're explosive, and they're carcinogenic, some of them. So lab safety is paramount. So before you even start going into setting up the lab, you need to clear some hurdles or set some things in place before starting the process. So lab safety equipment, this is not something I need to tell you about. If you ever worked in a lab, you know all about personal protective equipment. Make sure that you always wear goggles when you work with the synthesis reagents. It's not very much fun to get acetonitrile in your eyes. It burns. Other ones, I'm not going to even go there. Your clothing, you spill oxidizer on it, it's not coming off. D-block can take your fingertips off. The acid will burn you off. You're going to have peeling hands. Experience from many, many years ago when the safety in the lab were kind of lax and people didn't quite know about the repercussions of certain chemicals. So, fire codes. You will need to take in consideration the fire marshal requirements of your local state, of your local city. Make sure that you have a proper amount of solvent allowance for where in the building you are located. In some situation, better have chemistry floor on the first floor because it's the highest allowance. And it actually goes like this, the higher on the building you go. So if on the bottom you're allowed 20-liter container of flammable liquids, when you get to like fourth floor, it's only 200 milliliters. And make sure that you have all that information before bringing the instrument in, because it does use certain amount of solvent and generates certain amount of waste. So waste management. You want to make sure that you have set up your waste management for hazmat with company like Safe Harbors, for example. They do come and take your hazardous waste, they take it away and they burn it. So you want to make sure that you have that account set up before you start generating the waste from the instrumentation, because you have a full bucket of waste and what do you do with it? So they usually bring you the disposable drums and you go into them, like dump your waste into them, and then Safe Harbor comes in and takes them away to disintegrate. Reagent storage, again, don't have to go over. Flammable reagents or explosive reagents have to go into your flammable cabinet. You will have to have it. Controlled substances. As I mentioned before, AMA is a controlled substance. In some countries, acetic anhydride is a controlled substance. So In that situation, you would need to make sure that you have either DA approval, DA license to be able to purchase that. So, as I say, for your future needs in terms of laboratory setup and what kind of chemistry you're going to be doing, my kind of a little advice that I used to use in the lab: keep reagent of critical raw materials, place supply agreements, open standing orders, at least have 3 months supply if you're storage allowing for critical components, and keep reordering it at the 2 month mark, because in the past 3 years we had so much supply chain issues worldwide, delivery wise, production wise, etc. Keep the instrument replacement part to do small fixes. Instruments do come with the spare parts kit up front, but they do run out as the time goes on. So if, for example, you have a critical project and your valve dies and you don't have one, that will delay you for a couple of days until you get a new one. Please be in touch with us if you have any questions. Use the appropriate CpG porosity for your synthesis. Synthesis is a critical part of the process, so it's important to spend maximum time on it. Use fresh reagents when possible, because the certain amidites only stay on the instrument for a short period of time. Use specific CpG for specific length of the oligos. Define your workflow and write appropriate SOP. And as I say, quality system, you should start with that, with your safety, with your EHS, and make sure that you can actually have that in your facilities. So now I'm going to be finishing up the presentation and we'll be open for questions. Thank you so much.